Thank you for logging on. Good morning, and thank you for logging on. This is the Gene Therapy for Hemophilia B and Hemophilia A meeting of the California Technology Assessment Forum, and we'll be beginning in approximately 1 minute, so just hold on. Thank you.
Okay, we will start. Welcome, everyone. Good morning. Thank you for attending today's public meeting of the California Technology Assessment Forum, CTAF, a partner of ICER, the Institute of Clinical and Economic Review. I'm Rena Fox, chair of CTAF and of today's forum. Today, we will be discussing gene therapy for hemophilia B and hemophilia A, effectiveness and value. At the beginning of each meeting, we ask each of our CTAF panel members to disclose any financial relationships with industry. Every member of the CTAF panel who is present has met the ICER conflict of interest policy. Would each CTAF member introduce themselves, their work, and note whether they have any updates to this declaration? I will call on each CTAF member and I will begin. I'm Rena Fox, professor of medicine. I'm a physician and clinical researcher in the Division of General Internal Medicine at UCSF, and I have no updates to my conflicts of interest. I'm going to call on first Ralph Brindis.
Hello, everyone. Ralph Brindis, a clinical professor of medicine at UCSF, a cardiologist by training, and I have no additions and no conflicts of interest.
Thank you. Felicia Cohn. Felicia. Okay, we will move on.
Hi, sorry, I was unable to get off mute.
Okay.
I'm Felicia Cohn. I am the bioethics director at Kaiser Permanente, Orange County, and a clinical professor in the Department of Medicine at the University of California, Irvine School of Medicine. And no conflicts to report.
Thank you. Bob Collier.
Hi, I'm Bob Collier, 30-year patient advocate focused in the area of medical research with patient advocates in research, and I have no further conflicts at all.
Thank you, Bob. Sankeet Dhruva.
Thanks. Sanket Dhruva, cardiologist at the San Francisco Veterans Affairs Medical Center, assistant professor of medicine, and a health services researcher at UCSF, and I have no updates to my conflicts of interest.
Thank you. Kim Gregory.
Good morning. My name is Kimberly Gregory. I am professor and vice chair of at Cedars-Sinai Medical Center and at the UCLA School of Medicine. I'm the division and fellowship director of maternal fetal medicine and health service research, and I have no updates.
Thank you. Paul Heidenreich.
Hi, I'm Paul Heidenreich, a cardiologist and professor and vice chair for quality in the Department of Medicine at Stanford University. No conflicts.
Thank you, Paul. Jeff Klingman.
Hi, Jeff Klingman. I'm a chair of neurology at Kaiser Permanente in Northern California. No conflicts.
Thank you. Joy Melnikov.
Good morning, I'm Joy Milnico. I'm a professor emeritus at the University of California, Davis in the Department of Family and Community Medicine. I'm a family physician and a health services researcher, and I have no conflicts.
Thank you. Anne Roldo.
Hello, my name is Anne Roldo. I am an assistant professor of radiation oncology at UCLA Health. UCLA David Geffen School of Medicine, and I have no updates or conflicts.
Thank you, Anne. Rita Redberg.
Hi, I'm Rita Redberg. I'm a professor of medicine at UCSF and a cardiologist there, and I have no updates to my conflict of interest.
Thank you. Richard Seiden.
Hi, I'm a patient advocate and I have no conflicts of interest.
Okay, and Anthony Sowery.
Yes, good morning. I'm from San Francisco. I'm a volunteer for the National Patients Advocacy Foundation. I have no conflicts to report.
Thank you, CTAF members. At this point, we will also ask our individual patients and clinical experts to introduce themselves and declare if they have any potential conflicts of interest. That would be Brian O'Mahony, Mark Skinner, Dr. Margaret Rajni, and Dr. Miguel Escobar.
Good morning, good afternoon. My name is Brian O'Mahony. I'm the Chief Executive of the Irish Hemophilia Society, former president of EHC and WFH. And apart from those on the screen, I have no further conflicts.
Thank you. Mark Skinner.
Good morning, Mark Skinner. I am a patient. I live with severe hemophilia A. I am a former president of the National Hemophilia Foundation and the World Federation of Hemophilia. And the conflicts you see on the screen are related to my patient outcomes research, but nothing additional.
Thank you. Dr. Rajni.
That's Ragni. I thank you. I'm a professor of medicine and clinical translational research at the University of Pittsburgh, Department of Medicine, Division of Hematology Oncology. My conflicts have to do with my work on clinical trials with BioMarin and Spark, and also as an advisory board member and symposium speaker with Takeda. Thank you.
Thank you. And Dr. Escobar.
Yeah, I'm a professor of internal medicine and pediatrics at the University of Texas Health Science Center in Houston. I'm the director of the Hemophilia and Thrombosis Center. And my conflict of interest are related to my research that has been sponsored by some of the pharmaceutical companies, but I have nothing else to add.
Thank you, everyone. I will also add a reminder to everyone, whether it is later during the policy roundtable or during any public comment, that any individual making public comment should announce any relationship with industry or any other potential influence on judgment before you speak. We will also present this information on the policy slides. And at this point, I'm very happy to introduce Dr. Steve Pearson, president and founder of ICER. Steve.
Thank you very much, Rena, and good morning, everybody. So I'm just going to give a brief introduction to the meeting today, but first, welcome to you all. And I'd like to start by kind of framing why we're here today. Unfortunately, it's still not in person, but let's start with where we always do, and it's from the patient's perspective, their lived experience. So this is a mother of a person who's living with hemophilia B. And she said, one would think that once you're treated prophylactically, you're able to avoid future setbacks. Wrong. Not in the life of a person with hemophilia. There are still times when you need to drop everything to head to the ER to seek treatment. Living in a rural area where hemophilia is uncommon and there's a lack of knowledge of bleeding disorders makes seeking care difficult. Therefore, when we need to head to the ER, we call not only the Hemophilia Treatment Center, but also the pediatrician and the ER to give them our estimated arrival time. This is just one of many different comments, perspectives the ICER team gathered over the 8 months of this project, testifying, if you will, to the burden of this condition, the ways that people have to adapt their lives and live with the clinical as well as the broader effects. So we're here to talk about that. And in a sense, we're here because the day that treatments like the two that we'll be talking about today receive FDA approval, and now, of course, we're not 100% sure that that will be the case for both of these, but assuming that they will, in general, what happens is a sense of well-deserved celebration. again, we have conditions for which there's significant unmet need, affecting not just individuals, but families and others. And when there's a new treatment available, it represents a huge amount of work, investment, risk, the participation of patients and families in clinical trials. Sometimes things, fail late in the game. But when they do reach through to FDA approval, there's a great sense of joy, often, that we have new treatment options available for patients. Now, that's not the only thing that happens when FDA approval happens. There are still important questions. And those questions include questions about the evidence, because everybody knows that a certain evidence package is available at the time of FDA approval, but there are still almost always important questions about the risks and benefits, especially for particular types or individuals of patients. Other people will want to know, how do these new treatments fit into the evolving landscape? Are there other treatments coming next week? Are there other treatments already available that the FDA obviously didn't require a head-to-head trial? So these questions are part of the picture. Also, from the very first day, patients and others have to think about the price and the cost. The prices are set by the companies. In our country, there is no negotiation process at launch. And so there are real questions about what's a reasonable price and what will the costs ultimately be to patients themselves, but also to the health system and to the government and other payers. And we always want to think about what lessons we can learn, even at this early stage, to guide the actions in the future to make sure that we can do the best possible to create an innovative landscape that is ultimately going to be affordable. Now, that's why we're here today. But the other reason we're here today is that other things happen outside of the context, if you will, the immediate context of hemophilia. And this relates to the impact of new treatments, usually which add cost to the healthcare system. We'll talk about that. But we often think about words like affordability and sustainability as these kind of generic terms, but there are real people who are affected by these issues. And so I just wanted to bring a few of them into kind of closer view. And these are folks whose stories were highlighted in a wonderful article, if you want to read it, about medical debt in the United States. And the link is given below. But just as a couple of examples, again, Marcus and Allison Ward and their kids on the left fell into medical debt as the result of the premature delivery of the kids. And their insurance did not cover all the treatments that were necessary, and they had to move and change jobs in order to deal with that debt. Samantha and Ariane Buck in the middle there, they live in Arizona. And their story, again, a story of medical debt. It started with what they thought was going to be a short temporary zone without insurance when they couldn't afford it on their own between employers. And one of them ended up in the ER. And as a couple, they ended up with medical debt to the extent that they are now preparing to file for bankruptcy. Similarly, Sherry Foy there on the bottom right, she was on a kind of an employer's retirement health plan that had a cap on costs. And so when she unfortunately needed a lot more than that provided, she ran up bills of, I think, about $800,000 is what they talk about. And she fell into medical debt and bankruptcy. Now, the bottom line is that these are individual cases, but this survey that they did suggested that 100 million Americans are living in medical debt. And that means that they're either carrying expenses on their credit cards that they haven't been able to pay off yet, or they're formally in receivership with people trying to hunt them down. 100 million Americans. And of that number, 1 in 7, so I think that's about 15 million Americans said that because of their debt, they had been unable to go see a doctor or a hospital or go to the ER. They were turned away because of their existing medical debt. And about 50% of them said that they had self-rationed care, cut back, not taken pills, not gone to see the doctor, whatever it was, because of their medical debt. So we do have basically an epidemic of medical debt in this country, and it's often silent. But what we want to do is to make sure that we all recognize that part of the reason we're here today is because we need to come up with a system that balances that focus that we have and that we want to have on the individual patient and family. Today, it's treatments for hemophilia. And we want to, again, celebrate the innovation, talk about the new opportunities to help people with their lives. But we have to do that in a context in which We recognize that we all live within a system, a network, a community of other families who have other needs and who are trying to afford healthcare insurance and the out-of-pocket costs that go along with that. And we have to figure out how to do all of this together. So we're here to try to find that balance, to talk about our best way through it. You've already been introduced to the members of the CTAF. Let me tell you just briefly about ICER, the kind of hosting organization. First, in terms of our finances and potential conflicts, we keep an updated pie chart on our website of all our sources of revenue. And as you can see, the lion's share, about 69%, comes from nonprofit foundations. The primary ones within that are the Laura and John Arnold Foundation, also the California Healthcare Foundation, the Commonwealth Foundation, and others. There are small slices that you can see there, but we also take about 24% directly from health plans and provider groups or manufacturers of drugs. And that is kept separate for a policy summit program in which we bring leaders together to try to sort out some of these issues and create policy proposals looking forward. That money is not used to support the research or the meetings like the one today. So the report that serves as the foundation for the discussion starts, as I mentioned, about 8 months ago with scoping, as we call it, which is basically listening to patient groups, to clinical experts, to manufacturers and other stakeholders, just trying to say, what is the lived experience with this condition? What is the diversity of that experience? What do we don't know? Where are our blind spots? What matters most to patients about value? Where are the sources of evidence that we might not find in a medical journal? All of that we start off and we spend a fair amount of time before we even really go into the evidence. And that's what happens next. We do the ICER staff do an evidence review. We often work with outside academic health economists. In this case, we are welcoming Suri Walton, who's a professor at the University of Illinois, Chicago, who leads the cost effectiveness modeling effort. This whole report goes through several cycles, lots of touch points with patient groups and manufacturers, but formal comment and revision. And at the very end, we also have expert reviewers, two of whom are with us today, but they include Dr. Stephen Pipe, Dr. Margaret Ragney, and Mark Skinner, for whom we are, again, grateful that they were able to give us their input on the report. So what's in this report to support the discussion today? It is, in some ways, it all builds to a discussion around value. And we have a formal value assessment framework. And the final vote today will be on what we call long-term value for money. So what is that and how is the report structured to support that discussion? Well, first we have, if you will, 3 building blocks related to cost effectiveness, but it starts with the effectiveness. What can a treatment, in this case, a gene therapy, do to help individuals? Well, they can help us live longer. They can also potentially help people return to better function or have fewer side effects than other treatment options. And when we do think about value, we're obviously talking in some way about costs. And it's important to realize that we're not just talking about the drug costs in these reports. We talk about all of the costs in the healthcare system. So drugs that might have the ability to reduce the need for further testing or hospital visits, get that credit, if you will. Now, other pieces of this building block up to long-term value for money are equally important, but fall outside of a formal cost effectiveness model. But we'll talk about the benefits beyond health that's in some ways could potentially be measured, and in some ways they couldn't, but they may be important elements of value to patients and families. And then even more broadly, we'll talk about certain social or ethical priorities related to the severity of the condition, to the ability to address equity and other issues, that these are important framing elements in the ways that we think about value. So the ICER report has these structured elements in it, and they will all be featured in the discussion, the presentations for the public meeting today. So the agenda will quickly move to a presentation of the clinical evidence, followed by economic information. The CTAF will hear, deliberate, and we do have our patient experts and clinical experts at the table to help respond and to give their input as well. Then we're going to have a formal section on public comments with discussion with the CTAF. Now, these are all Pacific time. We will have a relatively early lunch break, Pacific time at around 11:15, and then come back at 11:50, where we will have further deliberation addressing the specific votes that we take on clinical effectiveness and value. Then, after a short break following the votes, we come back for an important policy roundtable where we will have, again, the patient experts, the clinical experts. We'll have 2 representatives from the payer community and 2 representatives, one each from the 2 drug, kind of the makers of these gene therapies. The goal of which is to reflect on what we know today as it's projecting forward. What will this all mean for drug insurance coverage? What will this mean about how insurers pay for it? Are there certain contracting options that are going to be on the table? What will clinicians and patients do in terms of information to guide shared decision-making? We'll cover all of that in a policy roundtable that will then end with final reflections from the members of CTAF and adjournment by 3 p.m. Pacific. So with that, let me hand it over to Dr. Jeff Tice, who will take us forward, starting with the presentation of the clinical evidence. Jeff?
Thanks, Steve, and good morning, everyone. Next slide, please. Oh, I'm going to swap over slide sets 1 second. So next slide, please. So I just want to acknowledge the hard work of many other members of the clinical team, Shahryar Fahim, Belen Hirsa Hagiwara, both researchers at ICER, and Dr. Janet Chu, an assistant professor at UCSF in the Division of General Internal Medicine, and none of us have any conflicts of interest to report. Next slide, please. So here to talk about hemophilia, both hemophilia B, which is a deficiency in factor IX, and hemophilia A, deficiency in factor VIII. Both illnesses are X-linked recessive, which means it's predominantly men who are afflicted. And the clinical experience is an increased tendency to bleed. There can be life-threatening bleeds, for instance, into the brain. But the most common kinds of bleeds that lead to disability, at least in the era of the availability of factor prophylaxis, is bleeding into joints and muscles, but particularly joints, leading to progressive joint damage, disability associated with the joint damage, and often joint replacement if that's available. Next slide, please. Prophylaxis with factor replacement has really changed the quality of life and length of life for patients living with hemophilia, but it's quite burdensome. It has to be given intravenously, and it has to be given frequently. Typically, in the past, 2 or 3 times a week. There are now some longer acting preparations, so patients can space it out every week or so. It's particularly challenging in young children administering IV therapy. And as patients age, both just some of the disabilities with aging, but also joint damage from living with hemophilia their entire life can make self-administration through IV ports challenging. And in part because of the challenges associated with IV therapy, adherence is an issue. So patients need to adhere to a regular schedule of administration to keep their factor levels high enough to prevent bleeds, and that doesn't always happen. Next slide, please. The main burdens are, continued risk for bleeding that limits patients' activities. Most patients are cared for at specialized centers. There aren't specialized centers in every community, and so access is an issue. Having access to factor therapy can be challenging sometimes, as the case that Steve reported. Some emergency rooms wouldn't have access to factor therapy and don't know how to manage patients with hemophilia who are bleeding, and because of the requirements for therapy and so on, there are often limitations on patients' time. This impacts their education, their career choices, their choices of recreation, where patients live, And there's a tremendous associated caregiver burden as well. So just want to acknowledge the enormous impacts beyond just the medical complications of hemophilia. Next slide, please. In talking to patients, they wanted to highlight that the annualized bleeding rates, which were the primary outcomes in the clinical trials, don't adequately capture all aspects of the benefits, burdens, and harms of prophylaxis therapy with factors. And that a curative therapy may be transformational in ways that even someone with hemophilia may not be able to really understand until it happens. So talking to some patients who've had gene therapy, or one patient who had a liver transplant for other reasons, but that cured his hemophilia, they just said life was so much better in ways that they really couldn't fathom before they were treated and cured, essentially, at least for a period of time. But they also note that insurance coverage for the specific factor therapy regimen that they need to maintain adequately high factor levels sometimes can be challenging. We certainly talked, we heard cases of children who are interested in playing soccer and other sports that had a significant physical impact, and they were requiring particularly high levels of factor and thus high doses of factor therapy to be able to do what they wanted to do, and there were often significant barriers to insurance coverage to maintain that level of factor therapy. Next slide, please. Okay, we're going to start with hemophilia B because ICER has covered hemophilia A in the past, and so we will get to hemophilia A in an update, but we're going to cover hemophilia B first. Next slide, please. So the therapy of interest, I put the full name up there, but we're all challenged at saying that. It's a mouthful, so everyone abbreviates the therapy, the gene therapy, as etranadaz. It's an adenovirus vector that delivers gene therapy to the liver. It's administered once, and it's only been studied in adults over 18, and it's been granted a priority review from the FDA, but as Steve said, it has not yet received FDA approval for therapy. Next slide, please. The population is adults with hemophilia B who don't have inhibitors to factor IX. So at this point, it hasn't been carefully studied in patients who have an immune reaction to factor IX, the so-called inhibitor. So patients without inhibitors who would be appropriate for routine prophylaxis with factor IX. So mostly severe or moderately severe levels of factor, but bleeding with that. And so on routine prophylaxis preventive therapy with factor. The primary intervention is etranadaz, and the comparator is prophylaxis with factor IX. There are a number of different factor preparations, but no other drugs available for prevention of bleeds in patients with hemophilia B. Next slide, please. So there's one pivotal trial, the phase 3 trial called the HOPE-B trial. It is a pre-post design, so 54 patients with hemophilia B on factor prophylaxis were studied for 6 months prior to receiving factor therapy to capture their current rates of bleeding on factor prophylaxis. They were then followed for up to 24 months. Mean age, just to give you some sense of the population, was 41 and a half years. And the primary outcome was the annualized bleeding rate at 12 months. So covering a period of time, what would the average bleeding rate be over a 1-year period, 12 months? They included patients 18 and older with moderate to severe disease who were on factor prophylaxis, and if they did have inhibitors to factor IX, they were excluded. Next slide, please. The primary outcome was the change in the annualized bleeding rate, comparing the bleeding rate from 7 to 18 months after treatment. So there was a 6-month period of transition while the liver received the gene therapy and then started to make factors. So they assessed bleeding rates in months 7 to 18 and compared them to the bleeding rates in the 6 months prior to receiving gene therapy. So each patient was compared to their bleeding rates in the period before gene therapy. And when you look at treated joint bleeds, which is one of the more common outcome measures, there was an 80% reduction in bleeding rates for patients after they received gene therapy compared to prior to gene therapy. For all treated bleeds, there was a reduction of 77%, and for all bleeds, those treated and not treated, there was a 64% reduction. So an improvement in bleeding rates, so that's a benefit in and of itself. And of course, they're no longer giving themselves prophylactic therapy. They no longer need to give themselves IV therapy on a regular basis. So another huge benefit to the therapy that isn't captured in the bleeding rates. Next slide, please. One of the concerns with this one-time therapy is what happens to factor levels over time. Do they continue to maintain adequate factor levels to prevent bleeds? In the HOPE-B trial, over the 18 months after therapy, the average factor levels were 39, 41, and 37. It's unclear whether this is a trend or just measurement variability over time. I also present the range. You see that some patients had much higher levels after treatment, up to 97 at 6 months and as high as 123 at 18 months. But some people had much lower levels, so 8 at 6 months down to 4 at 18 months. And that's the full range of patients in the HOPE-B trial. The next slide, please. There is also one phase 3 trial that has longer follow-up, in this case out to 39 months. You see it's only 3 patients. At 30 months, factor levels were actually higher than earlier levels, so up to 50. But at 36 months, back down to 37, but still maintained. This is only 2 of the 3 patients contributed data at 36 months, so that change may reflect different levels in the different patients. So pretty scant data beyond the 18-month period, but some data out to 36 months. That said, of these 3 patients, none of them had to go back on factor IX prophylaxis, so they all still remained free of the need to give themselves factor therapy. Next slide, please. So there were a number of harms. Again, this adenovirus infects the liver, and that can result in liver inflammation measured or assessed by liver enzyme elevations. And about 17% of the patients were treated with corticosteroid therapy to decrease the inflammation from the gene therapy. There are also a number of common side effects, headaches, flu-like illnesses, infusion reactions in 20 to 30% of patients, but these were all short-lived. There were a couple of deaths, one death from urosepsis that was assessed as unrelated to the treatment, and there was one cancer. Long-term development of cancer is a concern with gene therapy. In this case, they carefully genotyped the cancer and found no evidence of insertion of the factor gene into the cells that developed hepatocellular carcinoma. It was also much earlier than would be expected from gene therapy-induced cancer therapy, cancer causing effects on oncogenesis, but still a concern with gene therapy. I will say, just for context, this works not by inserting the gene into the DNA of the cell. It's a separate DNA fragment in the cell that then gets transcribed. So for the most part, you shouldn't get insertional damage to the DNA of cells, but it does happen in a small subset of cells. So this remains a concern long-term with gene therapy. Next slide, please. So uncertainties and controversies, normally for high quality of evidence, we'd like to see randomized trial with a long enough time period to feel confident that the benefits relative to standard therapy are durable. This was a single arm design, so there certainly can be selection bias. The kinds of patients who agreed to gene therapy in an experimental setting are probably different from the average patient with hemophilia B. There are very few patients treated to date. The pivotal trial had less than 60 patients. And when we're thinking about lifelong benefits, I mean, this therapy needs to continue to make therapy factors for the life of the patient. So there's the follow-up for looking at, do we maintain these factor levels, is relatively short. And finally, long-term harms are possible. The causing cancer, oncogenesis from insertional mutagenesis, is a concern. We don't know whether that's going to be a problem long-term. And similarly, there's clearly short-term liver injury, and there are some concerns that there may be long-term liver injury, and we just don't know yet. So there's some uncertainty here about the long-term harms of the therapy, as well as the long-term benefits. Next slide, please. The public comments received, the primary one for this was that there's sufficient certainty from the data about etranadaz to warrant either a B or an A rating. So this would be high certainty of evidence. And as you heard, at least the team's assessment was that the length of follow-up and the single arm trial design gave us enough uncertainty to warrant a different rating. Next slide, please. So in summary, clearly we had marked improvement in quality of life in most patients. They were able to live without factor therapy. They no longer needed to give themselves IV therapy, but there's still remaining uncertainties due to the single arm study design, the small number of patients treated, and the relatively short follow-up. One thing I haven't talked about yet, but is a concern, is once you've received gene therapy with this adenovirus vector, you develop antibodies to that virus, and so you can't receive any gene therapy that uses the same adenovirus. So you may be precluding future better treatments, at least using this vector. So there are some concerns among the patient community about taking a therapy now when it may preclude future therapies. And there's uncertainty about the long-term harms, including cancer formation. So we concluded in this case that there was moderate certainty of a small or substantial net health benefit. So we felt like the benefits seen at least were a small and potentially significant benefits compared to ongoing factor prophylaxis therapy, B+ rating. Next slide, please. With that, I'm going to turn to hemophilia A for a similar presentation. Next slide, please. The structure is similar. Valrox is the gene therapy delivering factor VIII therapy to the liver as a treatment for hemophilia A. It's given once, and the FDA has indicated they'll make a decision on March 31st of next year about coverage, or at least approval, excuse me. Next slide, please. The population is similar, but patients with hemophilia A without inhibitors who would be appropriate for routine prophylaxis. The intervention is Valrox, and we look at comparisons with both standard factor VIII therapy, but also emicizumab, which ICER reviewed in 2020 and actually found it to be dominant to factor VIII. So it was both more effective in reducing bleeds and less expensive than factor therapy. And this is becoming the standard therapy, though many patients with hemophilia, if they are stable on a therapy, are quite resistant to changing therapy, in part because of the harms of novel therapies in the past, leading to infections with chronic diseases and so on. So some patients continue to stay on factor therapy, but in general, young patients being treated with prophylaxis, starting treatment with prophylaxis, are often started on emicizumab now, rather than factor prophylaxis. Next slide, please. So there's, again, one pivotal trial, the GENERATE trial of Valrox. There were 134 patients, followed initially for 24 months, younger population than etranadez, so the average age was 31.7 years. A primary outcome initially was factor VIII levels at 12 months, but the FDA then rethought their requirements for the trials and required reporting of annualized bleeding rates at 12 months as an important primary outcome. Inclusion criteria, again, were adults on factor prophylaxis, and patients with inhibitors were excluded. Next slide, please. Bleeding rate reductions were 84%, both for treated joint bleeds and all treated bleeds. The reduction in all bleeds were not reported. Next slide, please. So one of the concerns with Valrox is declining factor levels over time. So here are the average factor levels from the pivotal trial, the GENERATE trial. At 12 months, the factor levels were, the average factor level across the patient population was 42.2, and at 24 months, that had declined to 24.2. And here we see there is also a range in patients. Some people get much higher levels. Some patients get much lower levels. Note, this is different than the etranadez range. There it was the full range. In this case, it's the interquartile range, so the 25th to 75th percentile. So we don't have the full range of patient experiences represented here. Next slide, please. We have much longer data from the phase 2 trial. So there are 7 patients treated with Valrox who have follow-up through 6 years. And in these 7 patients at year 1, their factor level was 64, and it declines steadily over time to an average of 9.8 at year 6. But again, 9.8 is still a reasonable level and prevents bleeds, and even many of the patients whose factor levels were below 5 or even below 1 did not resume factor prophylaxis. In the case of these 7 patients, none of them started factor prophylaxis through the 6 years of follow-up, so they remained off of IV, the requirement for IV therapy. Next slide, please. So the harms of Valrox, we saw a much higher level of corticosteroid use. So 79% of the patients were treated with corticosteroids for liver enzyme elevations, and the average duration was 230 days. So a significant period of time treated with corticosteroids. The other common side effects were headaches, nausea, arthralgias, and fatigue. There were 2 cancer diagnoses, one early on, an acinar cell carcinoma, so of the parotid gland. Again, careful gene analysis suggested that this was unrelated to the treatment. And then recently, 3 years after gene therapy, there was one case of acute leukemia. Preliminary analyses suggested that, again, that this was not due to insertional mutagenesis, though there's still ongoing work to really definitively say that. Next slide, please. And then briefly for context with emicizumab, you see that treated joint bleeds were not reported in the pivotal trial. Treated bleeds, there was a 68% reduction relative to a run-in period. So this, I should say, the pivotal trial for emicizumab is the Haven 3 trial. They had a number of different groups. The group D in the analysis of the paper of the different therapies, they had administration of emicizumab data. So again, patients on prophylaxis for a period of time prior to receiving emicizumab, so they could do pre-post comparisons. So this is similar to the kind of data that we have for Valrox. 68% reduction in treated bleeds, 63% reduction in all bleeds compared to prophylaxis with factor therapy. But we have no head-to-head trials, and the patient population was fairly different from the patient population treated with Valrox. There were fewer exclusion criteria. Age range was different. There were a number of different, many different differences in the patient groups. Next slide, please. So the uncertainties and controversies associated with Valrox, similar to to etranadaz, single arm design, so we worry about selection bias. A relatively small number of patients treated, follow-up relatively short, again, when thinking about lifelong therapy. And I think for Valrox, one of the things that's different is we clearly see declining factor levels over time. And the thinking is that at a certain point, many patients will need to go back on either factor prophylaxis or emicizumab. Physiologically, the target cell for gene therapy is hepatocytes, but factor VIII is normally made in endothelial cells as opposed to factor IX, which is made in hepatocytes normally. And there's some concern that this may explain why there's a fall off in time, because the target cells are not the cells that normally make factor VIII. And again, similar long-term concerns about cancer formation, oncogenesis, and liver injury, what may happen to liver cells over time, particularly making factor VIII, which isn't normally made in hepatocytes. So there's more concern about long-term liver injury with Valrox than there was for etranadaz. Next slide, please. A number of public comments on our assessment here and the clinical assessment. They noted that we had highlighted uncertainty about how many patients whose factor levels had fallen were on, had resumed prophylaxis. And it was actually the minority, and in an abstract presented at conference this summer, they had the numbers, and we included those in the full report. And they also commented that they thought that steroid use following therapy with Valrox would be lower in the real world, that in Europe we're not going to see immediate use of high-dose corticosteroids when there are small increases in liver enzyme values, but we have no real world data yet, so we don't know whether that in truth will be borne out. And certainly experts we talked to were uncertain about whether this would be the case. Next slide, please. So in summary, for comparing Valrox to emicizumab, there are no head-to-head comparisons. There were different inclusion and exclusion criteria, so the patient populations in the two studies were different. They did have similar reductions in bleeding events, proportionally a little bit more with Valrox than with emicizumab, but again, the patient populations were different. They have different adverse events, different burdens of treatment, so we really felt like we couldn't make a fair comparison between these two therapies. So we thought that the level of evidence was insufficient. There was too much uncertainty to really say anything meaningful about the relative benefits and harms of Valrox versus emicizumab. I should say emicizumab still requires parenteral delivery. It's an IV therapy, a subcutaneous therapy, but it doesn't require an IV port, needing to get into a vein to deliver the therapy, and can be, in general, it's given once a week, but it can be given dosed as infrequently as every 2 to 4 weeks. So there are some advantages compared to factor prophylaxis with emicizumab, but comparing the two, we really felt like there wasn't enough evidence to say anything with any degree of certainty comparing the two. Next slide, please. For Valrox versus factor VIII, certainly there are marked improvements for many patients over a period of years, but the decline in factor levels over time raises significant concerns about durability. Certainly, during the time they don't need factor prophylaxis, there's a marked reduction in the burden of therapy, but again, lots of uncertainty due to the single-arm study design, the small number of treated patients, and the relatively short follow-up. As with etranadaz, we worry about the fact that this precludes future, at least adenovirus-delivered gene therapies. And we worry a little bit more about the liver disease issue and similar concerns about oncogenesis long-term. Lots of uncertainty there. May never be a problem, but we don't really know yet. So in this case, we felt like the burden of harms was a little bit bigger. We felt like we couldn't give it a B+, but there could be substantial net health benefits as well. So we give this a C++ rating, so moderate certainty of a comparable small or substantial net health benefit. So greater uncertainty here, mostly because of more harm signals with Valrox and because of the rapid drop-off in factor levels. Next slide, please. So I now turn to just briefly the potential other benefits and contextual considerations. Next slide, please. So with current therapy, prophylaxis or emicizumab, the short-term risk for disability or death is low. Patients now with both hemophilia A and hemophilia B have a normal life expectancy, but they do develop significant disability from progressive joint disease. So we did hear about ongoing daily chronic pain from these from the joint damage and the impact it had on quality of life, on emotional health, and so on. We do think that gene therapy has the potential to give greater freedom to achieve goals, educational, family, work, and recreational. It certainly relieves caregiver burden of delivering IV therapy, and there's certainly reduced complexity of care after the first year. I didn't go into details about the first year, but there's a lot of careful monitoring during the first year of therapy and requirements for barrier contraception and many other things to make sure this adenovirus vector isn't passed on to potentially to children, to spouses, and so on. So there's a lot of monitoring the first year, but certainly after that, there's a great reduction in the complexity of care. Next slide, please. So with that, I can take questions on the clinical aspects of these two gene therapies prior to turning it over to the economic team.
Thank you very much, Dr. Tice, for that outstanding review of the clinical studies and clinical evidence. We have a little over 10 minutes budgeted for questions from any CTAF member, patient, or clinical experts. Please use your raised hand function and I will call on you. Lots of questions to start off with. Let's start with Joy.
Thanks, Jeff. That was very helpful and a good summary. I have a couple other questions just about the details of the therapy and the side effects. So the first one is just about failure, like what percentage of patients in these small trials just didn't work?
Yeah, so for etranadaz, it was 4%. So there were 2 patients who really didn't respond, and it turned out, the reasons: one had already had high titers of antibodies to the adenovirus vector, and so probably fought off the viral vectors before they could deliver the gene therapy to the liver. And then the second patient had a transfusion reaction during the infusion, and therapy was stopped, so they only got 10% of the dose. So there were 2 patients, so about 4%, who didn't receive who didn't have a response. The others did. For Valrox, I don't have the exact data. I don't remember exactly right after treatment, and maybe some of the experts in the room can do that. I know at 2 years, 5 patients who had factor levels less than 5 had resumed prophylaxis, and 1 patient who had a factor level over 5 resumed prophylaxis, presumably because of bleeding events. But the N is larger in that trial, so a similar, about a 5% of patients at that point.
Okay, thank you.
Can I just ask my follow-up question, and which is about the need for corticosteroid treatment, which seems to be sort of a significant thing. Maybe Jeff or one of the experts could just talk a little about what is the dosing required? What's the anticipated? I mean, is it really just initially, or then is it possible that people are going to require ongoing or repeated long courses of steroids?
Yeah, I'm trying to remember the exact. My recollection is that they often started with 60 milligrams of corticosteroids. I'm trying to find those numbers right now. Maybe one of the experts wants to.
That's correct. You're right on. Absolutely.
Okay. and
that's
so,
why you don't know about the outcomes in some of the studies, is because they were on steroids. So that sort of masked or slowed the loss of expression.
I think we're going to move to the next question, which looks like it is from Rita.
Thanks so much, Rena. Actually, I have 3 not very long questions, and just to save Jeff, because he has so much going on in his head, and that was a great presentation, I'll ask them one at a time. So first, Jeff, as we all know, randomized controlled trials would be the highest quality of evidence, and none of these, even though there are comparators and factor treatment, were randomized trials, and they were actually quite small and pre-post design, which seems very weak. Do you know why that was done? And are there any plans for randomized trials? Or
so I don't think randomized trials are planned. I think the thinking was there was no question that that there would be an effect of the therapy. But I'm not sure what the FDA thinking was in just requiring these therapies. It looks like David Rind may be able to weigh in a little bit more.
I oddly was at a meeting where this came up, where an FDA representative, this is a number of years ago, I know Mark was at this meeting as well. Basically, the FDA said that they viewed factor levels as essentially a perfect surrogate. And so, if you could normalize factor levels with a therapy, you knew what it was going to do to bleeding.
okay.
We do have about 3 or 4 minutes left, so I want to make sure a couple of other questions also. Yeah.
My other question was just if you could give absolute bleeding rates, because all the rates were relative risks. And the last was how reliable gene analysis is for cancers, and saying they aren't related.
Yeah, I mean, the absolute bleeding rates were low. I don't have them off the top of my head, but less than a couple of percent per year. I don't know. Again, maybe Dr. Aragni or Escobar want to chime in, and I can look that up and give you the absolute number. But the bleeding rates are low.
Yeah, the bleeding
so
rates
the pre-rates are low too.
The analyzed bleeding rates are comparable to factor VIII with ABRs between 0 and 1, so quite low levels of bleeding rates.
Yeah, that's what I was saying about, so 1% or less per year, so quite low.
The pre-rates, so then the reduction.
So then the reduction is 80, 60 to 80% from that. So the absolute difference is
but in
small.
absolute rate, you're talking 0.5% or something.
Yeah.
Anne, let's take the question.
My question was regarding the potential devastatingly secondary effect of malignancy. I was wondering if anyone could expand on what we know about the potential for a secondary malignancy, the timing, would it be, 1 year, 5 years, 25 years, 50 years? Basically, just any potential extra information we have about that.
I saw Dr. Escobar raise his eyes. He may want to say something on it. I think we just don't know. And I think it's more of a theoretical concern, but go ahead, Dr. Escobar. Or...
Yeah, I mean, so far, at least in all the clinical trials, we've seen 4 malignancies that they all seem to be unrelated. I mean, certainly it's completely unknown, what really is the risk, but the analysis that's been done in terms of insertion and mutagenesis in these tumors have been actually quite extensive, and they have not found that there's any relation between, the use of the vector and this malignancy so far. There's still some investigations ongoing, but there's no data to support that.
And I will say that they have looked at the background expected rates of cancer in the populations, and they are similar. But again, the numbers are so low, it's really hard to say.
Right. And the age of the treatments was low. It was 30 and 40 average. So 7 cancers in less than 100 people.
Yeah, the individual with the hepatocellular carcinoma, he had hepatitis B and C and cirrhosis. So those are all huge risk factors to develop this type of cancer.
All right, let's take the final question, Sanket.
Thanks. Quick question. What is the monitoring that's required as patients are treated with gene therapy? How often are they going to get, for example, LFTs? Is there other monitoring that may be associated with any treatment burden downstream?
I might turn to the experts because they have all the details in their heads quickly. I would be slow to get them all out.
These patients need to have liver function tests done really a couple times a week for several weeks thereafter, after the gene therapy, and then monitored long-term, a little less frequently, monthly, and then every few months. We do liver scans every 6 to 12 months. We see them, of course, during clinic, and we're monitoring for all the potential side effects, but including liver function abnormalities, cancer, other issues, but that's pretty much it. But it is very intense in the very beginning.
Thank you. As always, our CTAF members have outstanding questions. I'm sorry that we don't have time to get into detail with more of them, but I'm trying to keep us on time. So I'm now going to introduce Dr. Suri Walton, Professor, Department of Pharmacy Systems, Outcomes, and Policy in the College of Pharmacy at the University of Illinois at Chicago, for the presentation of the economic model. And there will be opportunities for CTAF members to ask questions following the economic model presentation. Okay, Dr. Walton.
Hi, can everyone hear me? Yep, great. Next slide. I want to start by acknowledging Ashton Maradi, a health economist at ICER, and Jyotimar Sarkar, a PhD student here at UIC, for their assistance with the modeling. Financial support was provided to UIC from ICER to cover our time. We have no financial conflicts to disclose for this project. Next slide. So there are 2 separate objectives in this project. The first one was to evaluate the lifetime cost effectiveness of etranacogene relative to treatment with factor IX in hemophilia B patients. And the second one was to evaluate the lifetime cost effectiveness of Valrox relative to treatment with imicizumab in hemophilia A. The next slide. Next. So in doing the analysis, we incorporated a couple ICER frameworks, the ultra rare framework and the single short-term transformative therapy framework. And the SST framework in particular has a number of scenarios associated with it. One of those scenarios is a $150,000 per year cap on cost offsets for the gene therapies. This scenario is going to be highlighted. It's also incorporated into the value-based price estimates. And this is because the cost of the comparators are so high, and I'll highlight that more later on. The model is a semi-Markov model. The setting is the United States. We took a healthcare sector perspective in the primary analysis. We have a scenario with societal perspective. It's a lifetime time horizon. We use 3% rate for discount, and the cycle length was 6 months. Next slide. So this diagram just shows how the model is structured. In hemophilia, there are acute impacts of bleeds that happen in the model each cycle that include costs and disutilities, but also there's a cumulative impact of bleeding on arthropathy. And we put this into the model. There's evidence-based connection between bleeds and Peterson scores. And so in the model, if you bleed more, it increases your transition across these Peterson scores. When you get to a Peterson score of 28, you receive surgery in the model. And then you return to a Peterson score of 14. And we do scenarios to, play with those assumptions. In the model, there's also mortality. That mortality rate is taken as just the average for US males from the CDC. It changes by age. The same structure is used in Hemo A, obviously with different comparators of Valrox and factor VIII. Next slide. So key assumptions in both models. First, in getting specific types of bleeds, such as joint bleeds versus non-joint bleeds, or treated bleeds versus untreated bleeds, sometimes we have to use proportional assumptions, and we take those proportions from all bleeds from data that has that kind of detail, and we use those same proportions across arms of the treatment. So the overall bleeding rates can change, but there's a proportional assumption on the different types within the overall bleeding. Another key thing that's going on is that gene therapy patients return to, they enter into the other arm when the efficacy of the gene therapy is projected to end. So we project factor levels across time. There's more detail later, but when the gene therapy stops working, patients go and start taking they go into the factor arm and start accumulating those costs and those bleed rates. And also there's no mortality effects of the treatments. Next slide. So in the Hemo B model, we take the bleed rates for etranacogene and factor IX from the HOPE trial. We also are projecting factor levels based on the limited data that we have across time in the etranacogene arm. And there's bleed rates associated with those lower factor levels from the literature with an adjustment that we use in the Hemo B. It actually involves adjusting bleeds related to factor VIII levels in Hemo A patients to try to provide a projected bleed rate as factor levels decline in Hemo B. When those projected bleed rates become larger than the HOPE trial in the etranacogene arm, we use those higher bleed rates. And then when the factor levels are projected to hit a level of 5% of the patients switch to factor IX. And when that level hits 1, all of the patients switch to factor IX. We have some scenarios to change that assumption, but that's the primary analysis. Next slide. So in the Hemo A model, we take the initial bleed rates from the generate data, and then we're going to switch over to the projected bleeds from factor levels when those become higher. In addition, in the early cycles for the first 4 years in the Hemo A model, it's assumed that 2% of patients are going to drop out each year. And we take the generate data and we have the 2% of patients with the highest bleed rates drop out of the data set. And then we calculate the bleeding rate from the remaining patients to use in those early cycles. And also there's a value-based contract that was suggested to us that we incorporate in the primary analysis. And when these patients exit, they receive a prorated rebate equal approximately to 4 years of prophylactic treatment. So the patients that drop out in year 1 sort of get the full rebate. The patients that drop out in year 2 kind of get 3/4 of that. And so that is incorporated for the 8% of patients that end up kind of immediately switching in the first 4 years. Again, when projected factor activity hits a level of 5% of the patients switch. When it gets down to a level of 1, everyone switches to imicizumab. And the bleed rates for imicizumab are taken from the Haven 3 trial using the group D patients. Next slide. So this shows the bleed rates used for etranacogene and factor IX. And as I discussed, there are also projected bleed rates for etranacogene. Once the factor rates become low enough and those projected bleeds become higher than this, then those get incorporated in the model. And then at the end, patients switch over to the factor IX and get those bleed rates and those costs. Next slide. So this shows the factor level projections that were used in the model. The circles are the actual data that we had. This blue line slipping down is the primary projection that we used. It's years on the x-axis. So you can see at sort of approximately 24 years, they reached that sort of factor level of 1 in the projections. If you look at that orange line, that was our optimistic scenario. As part of the SST, we do an optimistic and a pessimistic scenario of durability. So that's the optimistic scenario. If you look at the gray line, the steeper decline, that's our pessimistic scenario for projecting the factor levels. Next slide. So this shows our factor level projections in the Hemo A model. If you, again, the circles are the actual data. This convex blue line, which was a survival curve fit, was the primary projection for factor levels. In the optimistic scenario, we used this same blue line, but we made the bleeds a little bit lower at low factor levels prior to the switch. And if you look at the gray line there, that is our pessimistic durability scenario. Next slide. This shows just a table of the basis for projecting bleeds from factor levels. And again, these projections are used when they become higher than the initial, estimates that we have for bleeding. In the Hemo B model, we take these bleeds and multiply by 0.43 in the projections, but you can see that what happens is the bleed rates start to increase over time. When they get to a really low level, they are relatively high for a few cycles. But remember that pretty quickly, patients then switch over back to the factor arm. Next slide. This just kind of illustrates some of the bleed rates in the Hemophilia A model. The imicizumab bleed rates from the Haven D are constant in the model. Valrox in year 2, just to illustrate, has a relatively low bleed rate. This is taking from the generate data with having 2% of patients leave in year 1 and calculating the remaining bleed rates of those patients as I described. By year 10, there's a high bleed rate put in there because it's low projected factor levels between 3 and 1. But then again, patients are going to switch and the Valrox patients will end up with the imicizumab bleed rates later on. Next slide. This illustrates the utilities and disutilities used in the model. The utilities change by age. There's also a pretty significant disutility associated with the surgery state when they hit the Peterson score of 28. There's disutilities per cycle per bleed that are based on literature disutilities of bleed rates and assuming 2 full days of this disutility and 5 sort of half days of this disutility. And we have some scenarios that change those. Next slide. So here are the treatment costs used in the model. We have a manufacturer suggested price of $4 million for etranacogene. That happens once, of course, at the beginning of the model. In Factor 9, based on a market basket of products, we get an estimated annual cost of $688,000. That happens every single year. Because that's such a high cost, and at a cost of over $600,000 per year, at best, in terms of a value for health, you can only produce 1 quality per year at most. So the cost per quality being achieved is well above $600,000. And because of that, in addition to the primary analysis, we will be highlighting this $150,000 per year cap on costs. Next slide. So in the Hemo A model, we had a placeholder price of $2.5 million. Again, that happens once. We have as our comparator imicizumab. The projected cost per year in imicizumab are again over $600,000. And sort of for the same logic, we're going to be highlighting this $150,000 cost offset cap in addition to the primary analysis. Next slide. So here are just the per bleed costs that are in the model. There's some non-drug costs that vary by age that are literature-based that we use. And then there's also down below, you can see the drug costs for treating bleeds for Hemo B and the per bleed drug costs for Hemo A. There's also a societal scenario, and we had a per bleed societal cost that we used, again, from the literature there. Next slide. There's also some costs of arthropathy and surgery. So there's $650 per cycle costs. And then when you have surgery, it's a roughly $47,000 cost that's in the model. Next slide. So here are the results. Next slide. So in what we'll call the conventional analysis or the full cost offset analysis here, the drug costs associated with the Etranadez end up being about $5 million lower than for factor 9. And the total costs basically the same result there. There are somewhat fewer bleeds associated with the Etranadez and higher quality adjusted life years. So this makes Etranadez a dominant treatment. It costs less in total costs and it produces more qualities in this full cost offset model. Next slide. So in the Hemo A model for Valrox, we get that the total lifetime drug costs when you're taking the gene therapy is about $4 million less. You get slightly fewer bleeds and you get a slight gain in quality adjusted life years. This also makes, in this analysis, Valrox a dominant treatment. Next slide. So we conducted numerous one-way and probabilistic sensitivity analysis in the full cost offset model. In all of them, the gene therapies are less costly and have more quality, so that didn't change. We also conducted several scenario analyses. These included doubling the bleeding rates, having all patients switch at a factor level of 5 rather than 1. We had the optimistic and pessimistic duration models. We had patients start and return to a Peterson score of 20. We did a societal perspective. We had various other scenarios. And in all of those, except for the $150,000 cap model, the gene therapies were a dominant treatment. Next slide. So there's a lot of concern about durability in these products. So we did a special durability threshold analysis. What we did is we kind of went cycle by cycle and we had all the patients switch arms, and we found out how long would they have to stay in the gene therapy to become cost savings. And at the manufacturer's suggested placeholder price of $4 million for Etranadez, this happened at 8.5 years. So once the duration is over 8.5 years, the gene therapy looks cost saving in the full cost offset model. And in model 2 for Hemo A for Valrox, this happened after 4 years. Next slide. So this shows what happens if you do the $150,000 cost offset cap analysis here. And so we have a manufacturer suggested price of $4 million for Etranadez. If you cap the cost offsets at $150,000 per year in the model, you get a cost offset of $2.9 million. This leads to the incremental costs of the gene therapy being $1.1 million rather than cost saving. If you take the incremental qualities in the model, this gives you a price per quality of $1.8 million per quality. If you do the same thing in the Hemo A model, we have the placeholder price of $2.5 million. The cost offsets there end up being $1.97 million with that cap of $150,000 per year while they're on the gene therapy. And this leads to an incremental cost of $530,000 positive rather than negative. And with the incremental qualities of 0.1, this leads to a cost per quality of $5.3 million per quality. Next slide. So taking this $150,000 cap on cost offsets, we then went and calculated a value-based price. So a price at which the cost per quality would be, say, $150,000. That value-based price using this cost cap would be just over $2.9 million for Etranadez. Next slide. So this does the same thing. It caps the cost offsets at $150,000 and calculates value-based prices. And for Valrox, we get roughly $1.9 million. Next slide. So we received several comments. These included that the projected bleed rates are too high and the switching weights are too low at low projected factor levels. So we kept our base case as described. We did do a scenario where everyone switches at a factor level of 0.5, and at least in the full cost offset model, we got the same results. we acknowledge that there's imperfect information about exactly when patients are going to switch, exactly what the resulting bleed rates are going to be at those low factor levels. The Hemo A model should include factor VIII as a comparator. We heard that. We chose imicizumab, from a value-based perspective, because imicizumab was found to be a dominant treatment in prior analyses relative to factor VIII. So from a value-based price perspective and just overall value, we felt that was the more relevant comparator. There's not enough uncertainty projected in durability of the gene therapies. We did try to do optimistic and pessimistic scenarios, and we also did the durability threshold analysis to try to get at this, again, acknowledging imperfect information about what the exact durability is going to be. And finally, the models do not adequately project heterogeneity in patient responses. So these models are designed to estimate value for the health system for a population, and they're not meant to advise individual patients on treatment selection or capture all the potential patient heterogeneity. Next slide. So here's kind of the limitations or uncertainties and controversies. We had limited data on efficacy and duration of the gene therapies across time. The relationship between joint bleeds and surgery is admittedly imperfect. The model assumes one joint surgery at a time. It's likely undercounting surgeries in all of the arms. Many of the utility scores in the models come from patients with hemophilia. They may be higher than if you got the utilities from a healthy patient population speculating about whether they had hemophilia. The bleed data for both arms in model 1 for Hemo B come from trial data, which may differ from rates in actual practice. The bleed comparisons in the Hemo A model 2, are based on indirect comparisons across patient populations and settings. And finally, we have placeholder prices for Valrox and a manufacturer suggested placeholder price for Etranadez, but not actual prices. Next slide. So overall, we projected very large lifetime costs associated with both the treatments and comparators in both models. The gene therapies are projected to have large cost savings and small quality gains in the conventional full cost offset analysis. If you take the capped cost saving analysis of $150,000 per year, and you take the placeholder prices that we have, the gene therapies do not achieve common cost effectiveness benchmarks. And that is it. And I'm happy to take questions.
Thank you, Dr. Walton, for this work that you and your collaborators have done in your presentation. We have roughly little under 20 minutes budgeted for questions from CTAF members, patients, or clinical experts. And again, please use your raised hand function. Okay, let's start with Paul.
Oh, yeah, so thank you for that. And I had a question about the mortality, and it seemed that the assumption was that the life expectancy was similar to the US population, and that I guess there's no difference in treatments. And from my limited understanding, I thought that it was still thought that mortality was significantly lower than the US population. And I'm just wondering if that, and you may have mentioned that, but if we considered a significantly lower life expectancy for the group, and or potential differences in mortality, like if gene therapy was for some reason reducing bleeding, life-threatening bleeding, would the models be sensitive to small changes in life expectancy?
Well, yes, we use average, CDC mortality rates for US males in the model. And in our reading of the literature for patients that are being treated and staying on treatment, the mortality rates didn't look very different from the overall. And there's not enough evidence to know if there's mortality differences across the treatments. If there are mortality differences across the treatments, that would certainly impact the results. Small differences in mortality would likely not impact the results. Given in the full cost offset model, because the gene therapy is already dominant, additional mortality effects would just make it more dominant. And in the $150,000 cap scenario, it could alter the incremental cost per quality, and a big mortality difference would alter it by a lot, but I don't know the exact numbers there.
Okay, Brian.
Thank you. It was stated in the report that I think one of the assumptions was that if the factor 8 or factor 9 level respectively went down below 5%, 5% of people then would go back on prophylaxis each year. And if they went below 1%, all would go back on prophylaxis. I think you have to draw a distinction between A and B, because 5% level with factor 9 is probably much better, because factor 9 also distributes in the extravascular space. So you're probably going to get a better clinical outcome with 5% factor 9. And with factor 8, my understanding is that the chromogenic assay will not measure below 3%, so 3 IU per deciliter. So you really can't measure down to 1%. And the other point I'd make on this is that if you talk about 1%, that's an old measure, really. Certainly from the Council of Europe, in the EDQM, the recommended minimum trough level for factor concentrates is 3 to 5%, not 1%. So if an individual is on prophylaxis in Europe, the recommended minimum trough is 3 to 5%. And of course, if they're on emicizumab, they're getting a trough equivalent of 10 to 15%. So I think the 1% is certainly should not be looked at with any great seriousness. And the 5% has to be looked at carefully.
Let me jump in on that, because certainly we had lots of discussions about this. For the modeling purpose, we had to try to guess what patients would actually do who had received gene therapy and hadn't been needing prophylaxis. And we were suspicious that many of them might not follow appropriate guidelines to get their factor levels up if they hadn't started bleeding yet and had not needed prophylaxis for a long time. And Surrey certainly did scenario analyses to see whether it made a difference, but we certainly were making a guess about this. All we were trying to do was, there was nothing normative about the 5% versus 1% versus 3%. It was, what are we guessing patients might actually do in this setting? And obviously we don't know that we're right.
Yeah, well said. I mean, it's there just isn't good data out there on exactly what patients would do when they would switch. We can argue about when they should switch. And we had a primary analysis that we did in the previous report, and we kind of stayed true to that. And then we also conducted scenario analyses to see if it changed the conclusions, and it didn't change them. So that's the best I can say about that. And I'm not a clinical expert on when people should or would switch.
Rita.
Thanks. So my question relates to going over the quality assumptions, because they seem, besides the cost, obviously key to the paper, and the trials only measure bleeding rates. And so my understanding, I guess, was based on slide 60 that you imputed from the bleeding rates quality adjustments based on how often people would have to have surgery. Is that, can you just maybe tell us a little bit more about that and how reliable you think that was? I would say, I think actually putting a quality of life scale in the trials, in addition to doing randomized trials, would make a stronger design.
So there are a few features of utility in the model. First of all, there's an evidence-based utility gain. It's a small one, but there's a utility gain with the gene therapy relative to the factor arm. In the model,
and what is that utility gain based on?
I'm not. There's some surveys, EQ5D utility surveys, kind of pre-post in the gene arm versus pre-post in the factor arm, essentially, that suggest a small utility gain from the gene therapy. But importantly, in the model, there are disutilities that happen for each bleed. So every time there's a bleed in the model, there's a disutility associated with that. And then there is also disutility associated with surgery. That's happening less frequently, obviously. So the bleeds impact transitioning through the Peterson scores and lead to more surgeries if there's higher bleed rates, but there's also, more importantly, a dysutility associated with each bleed that's happening every cycle.
And they did, I didn't report these, and maybe I should have in the, but I couldn't put everything in the slide set, but there were clear quality of life improvements with gene therapy. Again, it's pre-post, so not randomized, but they did measure a number of different instruments, both the quality of life instruments as well as the UQ5D, as Dr. Walton indicated.
Right, it's pre-post and open label, so it kind of a little limited. All right, the other, well, go on. I'll maybe come back.
Okay, Kimberly.
Yeah, I just, are there any data or speculation about the potential like chronic use of steroids and the complications associated with that into the costs? And then also, what about the long-term liver surveillance that's likely to be needed? Is that, in terms of imaging, is that factored in, or do we have any? I know we talked about the immediate, but long-term, what would that look like?
Long-term liver considerations are certainly not in the model. We do include steroid use costs in the model, and we have a dysutility for steroid use in the first couple of cycles in the model. The main results in this model are so overwhelmingly driven by the cost of the drugs that it kind of doesn't show up in the results. It would take a pretty large chronic long-term liver problem to change the results.
Just to clarify in regards to the steroids, so far what we've seen in the trials is the use of steroids is short-term. There's no evidence that we need long-term use of steroids in these patients.
Yeah, we had short-term use of steroids in the model based on what we saw in the clinical, trial descriptions.
And by short terms, we mean 6 to 12 months. So not lifetime, but not a week either. I mean, so it is significant. And in the Valroff trial, they did report insomnia, Cushing syndrome. They explicitly reported some of the corticosteroid associated adverse events. And they're, yeah.
Could I just add to Kimberly's question? Serial monitoring for liver disease is baked into the hemophilia treatment model now because of the significant history of liver disease. Most of the participants in this cohort would likely have been involved in some type of serial monitoring of their liver disease anyway, so we shouldn't just assume that all of them would be new costs.
Bob.
Yeah, if you wouldn't mind, just for the benefit of all the non-health economists around, and I've seen this come up before, on slide 66 and 67, where we go into qualities and extended value life years, the drug cost is listed as multiples of the placeholder prices. And this is a comparison of a one-time drug gene therapy as opposed to ongoing factor therapies. So if you could speak to that, I'd appreciate it.
Sure, sure. The primary reason for the additional costs is because when the gene therapies reach low factor levels, patients switch to the comparator arm and they start accruing the costs, the drug costs from the comparator arm. There are also drug costs that are associated with the bleeds. But the primary difference of why it's 9 million, for example, instead of 4 million is because after a while they're switching to the factor arm and they're accruing those costs.
Rita, did you want to return to your previous question?
Thanks. I just wanted to be clear on whether you included the risk of cancers and how that was accounted for in the quality analysis.
There is not a risk of cancer in the model.
So purely an uncertainty right now.
Yeah.
So it wasn't modeled at all.
It was not modeled.
And do you think that, well, maybe it's for the clinical experts, how realistic that is?
I think that makes very good sense. It's more likely that our chronic hep C patients, if they're going to develop cancer, will develop it because of their hep C, as opposed to the gene therapy, at least with what we know now. Time will tell. I don't think we know that.
And their hep C,
Thanks.
and their hep C came from their factor use originally. It's a little hard to compare. Any other questions on the economic model? Okay, well, thank you again, Dr. Walton. And we are going to move now to the final section for the morning of public comments and discussion. We have 5 scheduled comments today. I will introduce the speakers and I will remind each speaker to declare any potential conflicts of interest before delivering their remarks. I'm going to start with the 3 commenters from industry, Dr. Debbie Benson-Kennedy, Dr. Wing Yen Wang, and Dr. Richard Ko. And we'll start with Dr. Benson-Kennedy.